Sorry not to be here with you today. It was a little bit difficult for me to join you at that time, and I'm happy that we can do that by Zoom. So we are leading here a lab that is working on metabolic liver diseases and their progression toward cancer. And 5 years ago, something like that, I was invited by Professor Gilles Mithieu to be an expert of the thesis of Monika that is here in front of you today, which I accepted with pleasure. And I was actually very impressed by Monika. So I proposed to her to join my lab after her thesis. And she did that in 2018 and start to work on the impact of some microRNAs in the development of these fatty liver diseases and cancer. And I guess that she's going to present to you today the last data that she got with MicroRNA 22. Now Monika has become, since the beginning of the year, maître assistant in my lab. And I don't know if you're aware, but in the Faculty of Medicine, these positions are reserved for people that are working actually part-time for the PI and part-time to develop a personal project. And Monika is actually pursuing what she did in her thesis, working half on the liver and half on the kidney. She was actually an expert in glycogen storage disease in the kidney and in the liver. And voila. So I'm very happy to have Monika in my lab, and very happy that you invited her to present her work. And now the floor, I guess, is yours, Monika.
Thank you. It should be better now? Okay. So first of all, thanks to the association for inviting me to do this talk. Thank you, Michelangelo, for your nice introduction. And as announced, I will be talking about microRNAs and their role in non-alcoholic fatty liver disease. Let me start off just by quickly introducing what non-alcoholic fatty liver disease is, or for short, we call it NAFLD. So NAFLD, as Michelangelo mentioned, is our main research interest in our lab, but more importantly, it is a globally spread pathology that is thought to be affecting around 25% of the adult population worldwide. So non-alcoholic fatty liver disease starts off due to different factors, the main one being obesity and metabolic alterations in the organism. It can also be due to sedentary lifestyle, so lack of exercise and poor diet. And in some cases, some patients develop NAFLD due to genetic predispositions, but it is a bit more rare. So the first stage of this pathology is the development of simple steatosis. So simple steatosis is just accumulation, excessive amount of accumulation of lipids into the hepatic cells. So this stage is still relatively reversible in most patients, because with some exercise and diets, they can go back to having a normal liver. However, if it is left untreated for on the course of several years, it can progress, and this stage leads to the development of NASH. So it's non-alcoholic steatohepatitis. So NASH is a more severe stage because it is characterized not only by lipid accumulation, but the patients have very strong inflammation and fibrosis development in the liver. But still, this stage can be reversed in some cases. It depends really on the extent of fibrosis inside the liver, but with better diet and exercise, it can still be reversed. And finally, in a portion of these patients, unfortunately, some of them can progress to having cirrhosis development. So cirrhosis is very often associated to alcoholism, but it can also occur in this type of pathology. So cirrhosis is much more serious, of course. It's characterized by lack of the decline of hepatic functions. The liver starts not working as properly. There is a lot of inflammation, a lot of fibrosis development inside of the liver, and we have a lot of cell deaths due to the injuries that occur in these livers. And because of these cell deaths, the liver starts to repopulate. It starts to have these regenerative nodules that are cells that proliferate much more to compensate for the cells that are lost. And all of these factors, so extensive proliferation, inflammation, and injury, can lead in these patients to the development of hepatocellular carcinoma. Importantly, as obesity is increasing worldwide, we have much more non-alcoholic fatty liver disease patients diagnosed, and we can see that in some of them, even when they're at this NASH stage, they can still develop complications such as hepatocellular carcinoma, and even in rarer cases, even with patients with only simple steatosis. So in our lab, we're interested to understand what are these molecular changes that occur at each step of the pathology, besides mutations, of course, that can make this disease progress from one stage to the other, and what makes hepatocellular carcinoma occur in some of these patients as well. So we are mainly interested in epigenetic changes, metabolic alterations, and also the alterations of non-coding RNAs. So today I will be talking about this specific type of non-coding RNAs, which are microRNAs, and how they can influence the progression of the disease. So if you have some questions during the presentation, please feel free to ask them. So what are microRNAs? Just a quick reminder. MicroRNAs, as the name indicates, they are very small RNAs, which are about 22, 3, 4 nucleotides long. They are transcribed as messenger RNAs in the nucleus. However, they are processed in a very particular manner. So I will not go too much into detail, but I will show you, for example, already in the nucleus, these microRNAs are cleaved on the extremities by this Drosha complex. Then this RNA loop is exported into the cytoplasm. It is processed again by dicer. And we obtain what we have, this mature microRNA duplex. So you have this 5' strand and 3' strand. And through this obscure process that we don't necessarily understand very well for now, one of these strands will be selected and will go into this protein complex that silences gene expression, which is the RISC complex. So the microRNA will serve as a template, and with the microRNA, the complex binds to microRNA response elements in messenger RNAs, and it blocks their translation. Either the target genes are being degraded or the translation is being blocked, but anyway, this results in the downregulation of the target genes of the microRNA. It's very important to say that one microRNA can target up to hundreds of genes at once. So it's not just one target, one microRNA. And in this way, microRNAs can regulate several different processes in the cell at once. Why we were interested in microRNAs and NAFLD? So NAFLD, as I mentioned, is a very is a predominantly metabolic disease, and we have already seen that there is an extensive amount of literature on microRNAs regulating hepatic metabolism. So as you can see here, they can regulate two main deregulated processes in the liver in NAFLD, which are glucose metabolism, so glycolysis, glycogen metabolism, gluconeogenesis, through which, so when glucose is there in an extensive amount, it contributes to NAFLD by this synthesis of lipids that will be accumulated in the liver. And these microRNAs can also regulate all of the factors that, all of the processes that are involved in lipid metabolism, such as lipid synthesis, lipid degradation, lipid import and export. So all of these processes which are deregulated into NAFLD. Today, I will talk about one particular microRNA that we have studied, and this is microRNA 22 and its role in non-alcoholic fatty liver disease. And we have chose to assess specifically this microRNA as it is one of the most highly expressed in the liver. There are thousands of microRNAs that are known so far that have been detected, but this one is one of the most highly expressed in the liver. And its particular role in non-alcoholic fatty liver disease on a functional level has never been established. And the second reason why we're interested in this microRNA so much is because the literature so far shows that every time we measure this microRNA in hepatocellular carcinoma, its expression is always lost. Its expression is always decreased compared to the non-tumoral part of the liver. So we have these two main questions. Can miR22 have a specific function in NAFLD before HCC development? And the second question, since microRNA-22 is decreased every time in tumors, could it be that it is a tumor suppressor in the liver? And we started off by first answering this, the first question, is miR22 involved in NAFLD progression? For this, we started off by some in silico analysis, because we had no idea if microRNA-22 can actually regulate hepatic metabolism. So for this, we just took some gene lists, curated gene lists through the Metacore database of genes that are involved in either lipid metabolism or glucose metabolism. And we cross-referenced this with a gene list that is bioinformatically generated of genes that are either predicted or validated to be targeted by this microRNA, to be regulated by this microRNA. And as you can see here, we had over 100 genes that are involved in lipid metabolism and are predicted or validated to be regulated by miR22. When we look at glucose metabolism, we had 20 of these genes. So it seems that miR22 has the potential to regulate hepatic metabolism and therefore influence NAFLD progression, but we needed to see if this is actually happening in the cell or not. So just an illustration, you can see here all of the genes that have the potential to be regulated by microRNA-22 that are involved in lipid metabolism and glucose metabolism. So this was very interesting, but it was only correlative studies, only in silico studies, and we wanted to know what would happen inside the organism, what would happen in vivo if microRNA-22 is no longer there. And for this, we developed a microRNA-22 knockout mice. So this means that we deleted the portion in the genome that codes for microRNA-22, and we saw what is happening inside on a whole body homeostasis already, if this would influence obesity or the general metabolic behavior of the mouse. And what you can see here is that when we deleted this gene inside of in the genome of the mice, when we compared them to control wild-type mice, there was absolutely no difference, which was very surprising, because you can see here through MRI analysis, the fat mass was the same, lean mass was the same. After sacrifice, we observed that even the white adipose tissues were exactly the same. Through CT scan here, which is represented, you can see in blue and purple, either the subcutaneous or abdominal adipose tissues were the same. So we could conclude that under normal conditions, the absence of miR22 had no seeming effect on metabolism, on whole body metabolism. However, when we took these mice and submitted them to a high-fat diet to induce obesity and NAFLD in the mouse, so for 3 months, these mice were fed with a high-fat diet, we could see that after these 3 months, the mice had significantly more fat mass compared to the control mice, which were also on a high-fat diet. After the sacrifice of the mice, we saw that both epidermal and mesenteric white adipose tissues were increased. And even through CT scan, you can see here the subcutaneous and abdominal adipose tissues were significantly higher in these miR22 knockout mice compared to control mice. So all the fat depots in the body, already on a whole body level, were increased in these microRNA-22 knockout mice compared to the control mice. So we concluded that under obesogenic conditions, the absence of miR22 exacerbates obesity, exacerbates lipid depots into the cells. So this is what happened on a whole body level, but we wanted to see if actually NAFLD is progressed or not in the absence of miR22. And if we look at the liver specifically, already here, you can see in the normally fed mice, there were no changes in the liver weight. The wild-type mice that were fed obesogenic diet had increased liver weight, but miR22 knockout mice had significantly bigger livers because they had more lipid depots into the liver. When we look at hepatic triglyceride content, so lipid content inside of the liver, already wild-type mice that were on a high-fat diet had increased levels, which was expected, but miR22 mice, knockout mice, had astonishingly much more lipids compared to the wild-type mice. And this was also observed on a histological level. So it seems that under obesogenic conditions, the absence of miR22 exacerbated also hepatic steatosis. And this was only after 3 months of diet. So it seems here that miR22 could be a brake a metabolic break that refrains the cells to accumulate too much lipids. And in the absence of this microRNA, you have no more this break, and you have more lipid depots in the cells. So this was interesting to see, but we wanted to know what is the exact molecular mechanism that leads to this exacerbated depots of lipids in the absence of miR-22, and how does miR-22 regulate NAFLD development. So in order to understand this, we did proteomic analysis in the livers. So we took the control livers and miR-22 knockout livers of the mice that were fed with a high-fat diet. And we did proteomic analysis. We detected almost 3,000 proteins, and 331 of these were differentiately expressed between wild type and miR-22 knockout mice. And half of them were upregulated. So we were mostly interested in these upregulated proteins, as since microRNA-22 is no longer there, all the target genes should be, most of them at least, should be upregulated. And we wanted to detect which are these direct targets of microRNA in the liver. So when we took these 148 upregulated proteins and we cross-referenced this list with the genes that were predicted to be targeted by microRNA-22 through in silico analysis, we observed that 54 of them did cross-reference in this way. And when we did enrichment analysis of what these 54 genes are doing, with no surprise, we observed that most of them were involved in metabolism. So it seems that miR-22 can specifically target lipid and glucose metabolism inside of hepatic cells. Just as a more in a more specific manner, as you can see here, we represented the glucose, the genes that are involved in glucose metabolism. Most of them were specifically involved in glycolysis, glycogen synthesis, and glycogen degradation. And when we looked at the lipid metabolism, there were genes that were mostly involved in lipid synthesis, fatty acid synthesis, fatty acid uptake, all of these factors that can actually contribute to NAFLD progression. So in this first conclusion, we can say that under obesogenic conditions, miR-22 loss can lead to a prostatotic metabolic switch, which can facilitate NAFLD progression. So that was the first question, but then we were wondering if this if microRNA-22 can also contribute to hepatocarcinogenesis in these mice. So as a reminder, as I said in the beginning, microRNA-22 is always decreased in HCC samples compared to the non-tumoral part of the tissue. And we were wondering if this microRNA could be actually a tumor suppressor inside of in the liver. So the first thing we wanted to know was whether all the publications that we saw so far about microRNA-22 being lost in HCC are we can confirm them, and indeed we did, because we took different data sets of cohorts of patients that had HCC, and we measured the expression of microRNA-22 either in the non-tumoral part of the liver and versus the tumoral part of the liver. And what we saw was that indeed in all the cohorts we tested, microRNA-22 was significantly decreased. And then when we looked at the survival of the patients in regards to the expression that they have of microRNA-22 in the tumor, we saw that the ones that present lower microRNA-22 in the tumor had worse prognosis. So microRNA-22 is systematically decreased in HCC, and it seems that these levels correlate with worse prognosis. Once we had this confirmation, we wanted to know if the microRNA-22 decrease actually has a functional role in the tumorigenesis, or if it's just, or if this downregulation is a bypass product of the tumorigenic process in the liver. And for this, we again turned to in vivo analysis with our microRNA-22 knockout mice. And here we induced tumorigenesis in either wild type control mice or microRNA-22 knockout mice. And we did this by injecting this hepatic carcinogen then. 2 months later, when the mice were adult, we put them either on a chow diet, a normal diet, or a high-fat obesogenic diet. And in the next couple of months, we were following the incidence of tumors in the liver. And interestingly, what we could see that already at 5 months after birth, these mice the wild-type mice on a normal diet and microRNA-22 mice on a normal diet still didn't have tumors. Neither did the wild-type mice on a high-fat diet. Okay. But the microRNA-22 knockout mice that were on their obesogenic diet already had tumor development in their livers. At 6 months, almost all of them had tumors, and some of them that were on a normal diet started developing tumors, as well as wild-type mice in obesogenic conditions. And these proportions were a bit worse at 7 months, but still wild-type mice without the obesogenic diet still didn't have tumors. So it seems that microRNA-22 absence, along with this obesogenic diet, accelerate tumor development in mice. And if we see the end result, this was at 11 months of age in the mice. Wild-type mice under a normal diet had tumors, not a lot, but still. MicroRNA-22 knockout mice, even without obesity, had more tumors compared to the wild types, and this was much more worse under obesogenic conditions. It is important to notice that in both groups of microRNA-22 knockout mice, either under obesity or not, they had much more aggressive tumors that were less differentiated compared to the wild-type mice. So we can conclude that microRNA-22 loss not only accelerates hepatic tumor initiation, but it also results in more aggressive tumors. Finally, as this microRNA is lost in hepatocarcinogenesis, we thought that rescuing its expression could be used as a therapy. And interestingly, even all of the cell lines of hepatic cancer that we have in the lab had almost no virtual expression of miR22. For example, here we have HepG2 cells that are very often used as a hepatoma model in vitro in the labs. And you can see that microRNA-22 is almost not detectable in these cells. So we thought we can transfect microRNA-22 mimicking oligonucleotides that will do this overexpression of microRNA-22 in the cell. And we see already how this affects the metabolism of the cancer. We did this in 2 types of cells. So we did HOH7 cells, which are hepatic cancer cells, and we did it also in AML cells, which are not cancerous, but they're liver cells that have been transformed, immortalized to be able to divide infinitely. So first we analyze glucose levels when we overexpress this microRNA in these immortalized hepatocytes. And to our surprise, we saw that this overexpression of microRNA-22 upregulated glycolysis levels, basal glycolysis and even compensatory. And when we did the same analysis in hepatoma cells, we observed similar results, but that were not that strong, possibly because already hepatoma cells have a very high glycolytic level. So it seems that miR22 overexpression induces glycolysis in both tumoral and non-tumoral cells in vitro. And finally, we analyzed fatty acid metabolism through this long-chain fatty acid oxidation stress test, in which we measure the oxygen consumption in the with or without blocking lipid usage in the mitochondria, so with this atomoxir drug. And what we saw was that in immortalized hepatocytes, when we overexpress microRNA-22, they become more dependent on fatty acid. And this effect was even stronger in hepatoma cells. So microRNA-22 overexpression renders HOH7 and AML cells more dependent on fatty acid. So as a second conclusion, the absence of miR22 led to accelerated and more aggressive carcinogenesis in vivo. Both in vivo and in vitro, microRNA-22 can modulate glycolysis and lipid metabolism. However, if we do want to use this overexpression in therapy, we need much more extensive analysis to see how it will affect tumoral and non-tumoral cells inside of the liver. So just as a brief summary of it all, for the first question, we can say that microRNA-22 indeed has a protective function against NAFLD progression from one stage to the other. And when it comes to hepatic tumors, microRNA-22 seems to have a hepatic tumor suppression function. So therefore, when it's lost, this accelerates tumor development in the liver. So I would like to thank you for your attention, and I'm open to any questions that you can have. I would like to thank my team. I would like to thank also the funding agencies. And please let me know if you have questions.
Thank you, Monika. That was so nice. And start for the mice, but maybe go faster, please. Any questions?
Before you are very clear and very organized presentation.
Thank you.
And I have a question. One last question: did you do some experiments in vivo using a mice?
No, we didn't. We didn't because in the liver per se, you don't need to overexpress it. I mean, maybe in NAFLD, but we haven't really thought about that. But what we were thinking about doing was to see if we can re-express it in vivo in the tumors. That would be really interesting, because even in the mice, I didn't show it here, but even in wild-type mice, when they develop tumors, microRNA 22 is lost. But for now, we still don't have the technology to target specifically the tumor in vivo. And that's the whole problem. That is why we wanted to test in non-tumoral and tumoral cells to see what will happen if you overexpress miR22, because if we target the whole liver, maybe we can also target the tumor and then decrease it. But this would be a really interesting approach. But for now, we don't have the technology to target specifically this.
And the second question: did you explore the mechanism why all the techniques are induced by the fees of MicroRNA? Or do you have any hypothesis why I did not use the fees of MicroRNA?
We are not sure yet. The reviewers did ask us that. We are not sure yet, but what we hypothesized is that by analyzing the promoter of the microRNA, etc., what we think is that some specific lipids that accumulate during NAFLD inhibit the expression of the microRNA. We don't think it's like a transcription factor that is not there, but I think it's some metabolites that influence the promoter of the microRNA, but we didn't analyze this specifically, but it's probably the lipids.
Instead of testing on cells, would liver organoids be a model system to test?
Yes, of course. We are currently, in the last couple of years, we were developing this in the lab. So what we're doing is taking embryonic stem cells or induced pluripotent stem cells, and we're trying to develop HLOs, or human liver organoids. And this seems to be working, because we have really nice organoids that have hepatocytes and Kupffer cells and stellate cells, but we're still kind of in the process of characterizing them. But we have already tried to transfect them with microRNA, so not specifically miR22, but we tried this with another microRNA of Marta, which is I think there because she's doing all of this. But it seems to be working, because we managed to transfect them, we managed to detect the microRNA that is there, and we managed to see some changes in either steatosis or fibrosis levels, because these organoids, we can clearly now use them as a model for NAFLD, because we treat them either with oleate or palmitate or both to induce steatosis. We can put some cytokines that induce inflammation, just like in a human liver. And we are currently trying to do this. So we're having some nice results, but it needs a lot of characterization. So I don't know if somebody's working with organoids, but it also costs a lot, but a lot. So we cannot just play around. But yeah, it's a really interesting point. Yes?
So you saw that cancer patients.
This we did also analyze at different stages of NAFLD, how this microRNA is expressed. And it was really cohort dependent because we had, I think, 2 cohorts when it was downregulated. In one cohort, it was unchanged. In another cohort, it was upregulated. So either there is a technical issue there with the data sets, or maybe it's really dependent on the population that you're analyzing and their lifestyle habits and the level of progression of NAFLD. So for now, we couldn't really conclude on that.
So these mice were, so they were the normal exercises.
So what we had a cohort, I didn't show this here, but for example, we had a cohort where we put them on high-fat diet. They lost, because mice, we did see this decrease even on NAFLD stages already with the fat accumulation, they start losing microRNA expression, 22. And in one of our cohorts, we saw this decrease. And then we had a third group of mice that were on a high-fat diet, but were doing exercise. And there was not a complete rescue, but we could see some small upregulation. So we didn't know how physiologically relevant this upregulation is, so it's a very complex matter. But I think with some dieting, it actually, the expression really follows the state of the liver. If you have too much steatosis, especially in mice, we see that it is lost. But then if you start fixing it, it also goes up. So I don't know if I'm being clear.
No question. Well, if there are no questions, maybe we take discussion pizza. It's not a low-fat diet, but But thank you very much, Monika, for coming over and all the way from Syria and start also to start the relationship between the NP and CN. It's very nice. So yeah.
Well, thank you for inviting me.
And then we go for pizza.
Thank you.